Hello, dear participant. We are in SWAM course of Introductory Animal Physiology. Our module is Digestive Physiology of Monogastric. Course title is Permeability Characteristics of Intestine and Forces Governing in the Absorption. Hi, I am Dr. Manish Kumar, Assistant Professor, the Department of Veterinary Physiology and Biochemistry, Faculty of Veterinary and Animal Science, Institute of Agricultural Science, Banaras Hindu University. In this topic, we will be covering movement of the particles across the cell membrane, diffusion, active transport across the cell membrane, osmosis, pinocytosis, phagocytosis, paracellular transport, and transcellular transport of the nutrients, and absorption of the water and electrolytes. Let's start with the movement of the particles across the cell membrane. So now we have just in the last class we have seen how that nutrients are being digested and absorbed. Now this absorbed nutrients need to be transported through the cell membrane to the site of utilization for the different function. So absorptive epithelial cells lining the intestine have an apical membrane in contact with the luminal content and the basolateral membrane in contact with the extracellular fluid. The apical membrane of the enterocytes has microvilli that project into the lumen to increase the surface area for the absorption. These cell membrane are composed of bilayer of phospholipids. Phospholipids have the hydrophilic head extending into the water on the outside and inside of the cells a hydrophobic region between the two hydrophilic surface. Interspersed among the phospholipid are cholesterol molecules and various type of protein. These proteins are often glycoprotein and they can extend varying distance in or out of the cell membrane. Some are important as receptor for the hormones or neurotransmitter, also serve as a membrane channel to allow or facilitate the entry of the material across the lipid bilayer. Lipid-soluble hydrophobic compounds can easily cross the lipid bilayer of the cell membrane. Water and hydrophilic material cannot easily pass the lipid bilayer without some accommodation in the form of protein-based transport. Like all the cell membrane, a thin layer of glycoproteins, oligosaccharides and glycolipids cover the lipid bilayer to form the glycocalyx. A thin layer of the water, the unstirred water layer, adhere to the glycocalyx by surface tension forces. The unstirred water layer and glycocalyx do not impede the absorption of water-soluble solute but do form a barrier to entry of large lipophilic substances that might ordinarily cross the apical membrane unimpeded. The bulk of absorption is performed by the villous absorptive enterocytes in the small intestine. Absorptive enterocytes also exist in the large intestine, but they lack many of the enzyme and transport molecules of the small intestine villous enterocytes, which limits what they can absorb. Now coming to the diffusion. Diffusion is a process in which the particle in solution move from an area of high concentration to the area of low concentration. Particles will also move from one compartment to the other compartment if the membrane which separates that two compartments are permeable to those substances. Facilitated or carrier-mediated diffusion, solutes that are too large or which carry the charges can utilize the carrier protein to facilitate their diffusion across the cell membrane. These carrier proteins recognize very specific molecules and form a channel of low resistance that allow the molecules to move down the electrochemical gradient to enter the side of the membrane. Often these carrier proteins are regulatable. They may be under the hormonal or neuronal control. In some cases, ions will be transported together using transporter. This involves a transporter that facilitates the movement of one ion against its electrochemical gradient. By pairing with the another ion is moving down its electrochemical gradient. This strategy is repeatedly used to absorb the material across the intestinal cell membrane. Sodium is normally in much higher concentration within the lumen of the intestine than inside the intestinal epithelial cells. The amino acid aspartate has the negative charge and is too large to cross the cell membrane, even though it will also be in much higher concentration in the lumen of the intestine than inside the epithelial cells. Since the inside of the cells is negatively charged compared to the outside of the cell, sodium will have both electrical and the concentration gradient pushing it to move into the cells. Aspartate will be moving into the cell down its concentration gradient but against its electrical gradient. Carrier protein in the apical membrane of the intestinal epithelial cells recognize aspartate and sodium and can open up when both are bound to the carrier protein. The combined electrochemical force supplied by the sodium moving down its electrical and concentration gradient and aspartate moving down its concentration gradient will help drive the aspartate molecules across the membrane against its electrical gradient and through the membrane despite its large size. Now coming to the active transport across the cell membrane. Active transport means it is in this type of transport, energy is being utilized in the form of ATP. So active transport is usually required when moving the substances against its electrochemical gradient, and it is also called as pump, and active transport will create the concentration gradient very high, and this will be very effective, and it is a very selective for the transport of the ions. An example of such pump is electrogenic sodium potassium ATPase pump. This pump keeps the extracellular sodium concentration high and intracellular potassium concentration high. It also generates an electrical potential difference across the cell membrane, keeping the inside negative relatively to the outside of the cells. Now coming to the osmosis. Osmosis, a critical concept in the biology, is the concept of osmosis. Water will always try to move from an area of or compartment of low solute concentration to an area of high solute concentration. Each ion or particles within the solution act as osmotic particles regardless of its charges. The osmolarity of the component or solution is determined by concentration or number of moles of particles in that solution. The key to absorption of water across the intestinal tract is to absorb solute. Pinocytosis. Pinocytosis is the method by which the cells absorb a small particle outside the cells and bring them inside. The word pinocytosis comes from the Greek, the cell drinking. During this process, the cell surrounds particles and then pinches off part of its membrane to enclose the particles within vesicles, which are small spheres of the membrane. This process is usually for taking extracellular fluid. In some circumstances, very large or highly charged particle can be moved across the cell membrane via the process called the endocytosis. The substances in the pinocytosis, substances to be transported come in contact with the cell membrane, and an endocytic membrane form around the substances, and it is taken into the cell as a membrane surrounded vesicles, and very often the membrane will cross the membrane on the opposite side of the cell by exocytosis. This process is used to absorb the immunoglobulin, a very large protein in the colostrum that provides the passive immunity from mother to the neonates. Now that we will be talking about the paracellular absorption. Paracellular absorption means that the cells will unite together, and from all the sides they will form the tight junctions. So in between the two cells, if anything is being transported, it is called as paracellular. The tight junctions are composed of several proteins that forms a seal between the cells that relatively impermeable to the bacteria, virus and large molecules that have been ingested. The tight junctions also provide resistance to the passage of the small ions and water. However, the resistance can be overcome if the electrochemical force driving the ions to the opposite side of a tight junction are great enough. Water channel and ion channels do exist in the tight junctions. They provide the path of lower resistance for water and ions that are utilized only when there is a great differences in the osmolarity or concentration on one side of the tight junction. Movement across the tight junctions is only significant for a smaller ion such as sodium, chloride, potassium, calcium, phosphate, etc. Absorption of solute across the tight junctions between the endocyte from the lumen directly into the extracellular fluid is referred to as paracellular transport. Now coming to the transcellular transport. Transcellular means the molecules are being transported through the cells. Most of the nutrients of the body are too large to cross the tight junctions and must be moved across the absorptive enterocytes of the villus by a variety of transport mechanisms. Transport proteins can facilitate passive diffusion or allow active transport, that is pumping of the solute against electrochemical gradient at the expense of ATP. This process involves moving solute from lumen to cytosol of the enterocyte across the apical membrane and movement of solute from the cytosols to extracellular fluid across the basolateral membrane. The mechanism used to transport a solute across the apical membrane is often quite different from that used to transport the solute across the basolateral membrane. So both the mechanisms are different. Here in the diagram you can see this is the X and this is the Y, and this are transported across the intestine transcellular mechanism. They have to cross both the apical and basolateral membrane. This is the apical membrane and this is the basolateral membrane. These mechanisms are efficient even if relatively low amount of the solutes are present in the lumen compared to the extracellular fluid. The Z line, the ion Z traveling tight junction and is absorbed by the paracellular process. The paracellular transport is concentration driven and only functions when the solid's concentration in the lumen is far higher than the ion concentration in the extracellular fluid. Absorption of dietary minerals and secreted electrolytes. Dietary electrolytes, sodium, potassium, chloride and minerals must be in solution within the fluid of the digestive tract if they are to be absorbed. Many minerals that might be in an insoluble form within the diet component become soluble after exposure to the acid in the stomach. The stomach can absorb some sodium and potassium. For monogastric species, bulk of minerals and electrolyte absorption occurs in a small and large intestine. Colon can also absorb electrolytes quite well if they are still present in the lumen. So this is the major mechanism utilized for the absorption of electrolytes, that is hexose, amino acid and water in the various segments of the intestinal tract can be seen here, that in the duodenum how the different particles and different ions are being absorbed, and then upper jejunum, middle jejunum, lower jejunum, ileum and colon. Now we will be talking about the control of intestinal transport and electrolytes and water. Absorption of water. The two forces are at work to move the water. Osmotic pressure is generated by the movement of solute from one compartment to the another, creating an osmotic gradient. Hydrostatic pressure is a physical force created when water is moved into a confined area. Aquaporine channels exist in apical and basolateral membrane of the enterocytes that allow water to move across the lipid bilayer of the cell membrane to follow the osmotic gradient. These aquaporine channels do not allow any charged ions through them. Solute is typically absorbed across the apical membrane and crosses the basolateral membrane to enter the lateral space between the epithelial cells. This draws the water into the lateral space between the cells through the aquaporins. As more solute and more water enters this confined space, it increases the hydrostatic pressure. Distension of the elastic cell membrane of the lateral space also allow the hydrostatic pressure to increase. Tight junctions offer more resistance to flow of water than the fenestrated endothelial basement membrane of the capillary bed. The tight junctions are not impermeable to the water, just less permeable than the capillary endothelium. Water channel formed by claudin protein in the tight junctions generally resist the flow of water, but this resistance can be overcome if osmotic force are great enough on one or another side of the membrane. The factors involved in water and electrolyte transport. The transport of water and electrolyte by intestinal mucosa involves the several types of the cells and a structural relationship. The important components of this process are epithelial cells. Epithelial cells represent the largest population of the cells of the intestinal mucosa, of which there are four major types. They are columnar cells, mucosal endocrine cells, goblet cells and Pence cells. As the cells reaches the villus tip, they undergo the apoptosis and are sloughed up. This we have seen previously also, that how cells will go up and it will change their characteristics. As the cells undergo the crypt to the villus, significantly changes in cellular morphology are evident, characterized by increasing polarity of the cells, differences in the cellular organelles, compartment, development of the microvillus membrane and terminal web, and alteration in other cytoskeleton and tight junctions structural features. The latter are accompanied by the decrease in the junctional permeability, a property that may rise out of necessity to achieve the efficient absorption of nutrients and electrolytes with minimum of the backflux. The regional differences in tight junctions permeability are reflected by number of intracellular strands that make up the tight junctions complex. The number of strands in this anastomosing network in villus cells is much greater than in the crypt cells, making the villus region more impervious to passive diffusion of the water and electrolytes. Now coming to the blood capillaries and lymphatics. The blood capillaries and lymphatics have a major role in the intestinal water and electrolyte transport. During absorption, they rapidly remove absorbed nutrients, water and electrolyte from the interstitium, thereby allowing the bacterial transport to proceed. Similarly, active secretion of water and electrolytes is accompanied by increased mucosal blood flow and capillary filtration, and by decreased venous lymph pressure and total lymphatic flow. In each case, in order for the efficient absorption and secretion to occur, the events of the mucosal transport, capillary flow and lymphatic functions must be coordinated. The neuronal and hormonal agents play a major role in the integration of these events. Intestinal motility, this also helps in the absorption or transport of the molecules. Coordinated pattern of the intestinal motility ensure the mixing and propulsive activity of luminal content proceeds appropriately. This mechanical activity can significantly enhance the absorption of nutrients, electrolytes and water through the several potential mechanisms. That segmentation contraction takes place, which regularly mix the food kind or food particles, and it comes in contact with the mucosa and nutrients are being absorbed. Mucosal cell involved in the regulation of the gut and water electrolyte transport. Several peptides, active amines and other agents prepare to modulate the intestinal fluid and electrolyte transport. Mucosal endocrine cells most likely regulate the intestinal ion transport functions through the paracrine action. Mucosal endocrine cells probably serve two important functions relevant to the control of intestinal water electrolyte transport. First one is that they provide the means for the mucosa to sense and rapidly respond to the alteration in the luminal content and milieu, stimulate the release of hormonal peptide and active amines, causes the appropriate changes in ion transport functions, blood and lymphatic flow and intestinal motility. Secondly, they may function as a fine-tuning mechanism or amplifier for the modulatory signals received from the neuronal sources. So, water balance in the gastrointestinal tract of a dog, for example, if you see that total water intake from the food or water drinking is around 600 ml, and different secretions are there. Salivary gland secretion 300 ml, gastric juice 600 ml, like this bile, and all together it water is around 3050 ml, and it is being absorbed around 3015 ml, and only 35 ml of the water is excreted in the feces. So that means only about 1% of the water is exit the tract with the feces. In the horse also, if you will see, the total consumption of water is around 144 liter and 136.5 liter are being absorbed, and only 7.5 liter of water is coming in feces per day. So in this we have seen that how the different molecules or ions are being transported and are absorbed, and what are the forces governing, what are the different channels are there, and we have seen that digestion of the food and the different digestive tract. So with this we are completing the digestive system of the monogastric animal. Thank you.